Good morning, everyone, and thank you very much for the kind invitation. Can you hear me? And can you see my slides as well?
Yes, very well.
Thank you. Thank you very much. I'm really happy to participate in the SARE Congress. I would be more happy if I was able to join face to face. I hope we'll manage it next year. And thank you very much for the kind invitation and introduction. Here are my conflicts. So we know that 20 to 40% of patients with COPD have an eosinophilic phenotype, meaning that they have ongoing type 2 inflammation. And this inflammation is the target of biologics targeting interleukin 5 and interleukin 4 or 13 pathways. And more importantly, biologic targeting alarmins are considered to have broader effectiveness as they target upstream mediators of inflammation, potentially impacting both the type 2 and non-type 2 inflammatory pathways in COPD. Over the last years, we have an increasing number of phase 3 trials looking at biologics, and we'll get into a bit more detail in the design of this trial during this presentation, because as we know, some of them were positive, some were negative, and we need this information in order to be able to interpret them. So we'll start from the dupilumab, that was tested. Dupilumab, which is a antibody that inhibits interleukin 4 and 13 and inhibits the alternative T2 inflammation pathway. And it was tested in two large trials, the Boreas and NOTU study, each recruiting around 930 participants. Both these trials had very focused eligibility criteria. So they selected patients with high blood eosinophils of at least 300 cells per microliter, patients with a history of exacerbations, at least two moderate or one severe in the previous year, and also patients that were already receiving a triple combination of a LABA, LAMA, and an ICS. And importantly, they capped their the current smoker participants at 30%, because initially there were concerns about whether dupilumab is equally effective among smokers and non-smokers, although no significant no significant difference was observed among these two groups. Now, both Boreas and NOTU showed a significant reduction in the rate of moderate to severe exacerbations of around 30%. And what is interesting is that while they did show prolongation of the time to first severe exacerbation that reached significance, the pooled data of both trials did not manage to demonstrate a significant reduction in the rate of severe exacerbations. And the reason for that is likely that both trials started during the COVID pandemic, and at the time patients were shielding and they were avoiding attending hospitals, and probably we know that the rate of severe exacerbations of patients were lower than usually. Both Boreas and NOTU showed benefits with regards to FEV1, and there was some benefits in health status, but only in Borea study we had a significant improvement in the number of responders to dupilumab. There were no safety signals. An important subgroup analysis that was presented in the dupilumab studies was the study looking at high versus low FeNO, and we can see, as expected, that patients with higher FeNO responded better to dupilumab, and FeNO is an indicator of the alternate inflammation pathway being active, so it's only logical that people with higher FeNO responded better to dupilumab. Benralizumab, we're moving now to benralizumab, that was assessed in two large trials, the Galathea and Terra Nova trials. We can see that the study populations were high. We can see that they both recruited patients with an exacerbation history, at least two moderate or one severe exacerbations. They were a bit more flexible and they allowed dual bronchodilators or triple combination as a baseline treatment of patients. And they did not have an eosinophilic threshold. And we can see here that both Galathea and Terra Nova did not show a very significant, clinically significant benefit in the rate of moderate or severe exacerbations. There was only a borderline benefit in the Galathea study, both for moderate and severe exacerbations, while there were no signals on the other outcomes either. Interestingly, in this very well conducted post hoc analysis, we can see that if we select the patients carefully, so patients with high eosinophils, with history of exacerbation, who were already receiving triple therapy at the time of recruitment, benralizumab did show benefit in reduction of exacerbation rate. So we can see that, as we said, selection of participants is very important. More recently, we saw the results of the ABRA trial, which was a phase 2 trial that was conducted by Mona Bafadel's group and recruited acute exacerbations of asthma or COPD with high blood eosinophils. And I will focus on the COPD part of the study. It only recruited 51 patients with COPD and without coexistent asthma. But even in this population, a single dose of benralizumab during the exacerbation, with or without prednisolone, appeared to reduce treatment failure at 90 days quite significantly. So the relative risk was 0.43, with confidence intervals from 0.20 to 0.93. And of course, we have another trial coming, the Resolute trial. That is a phase 3 stable trial that selected patients with higher eosinophils, so stricter criteria, and we'll hopefully have the results soon. When moving to mepolizumab, it was assessed in two trials, the Metreo and Metrex. And we can see that the Metreo trial only recruited patients with high eosinophils, while the Metrex included both eosinophilic and non-eosinophilic patients. The eligibility criteria, again, we had the history of exacerbations. We have a background high-dose ICS used during the preceding year. And I also already discussed about eosinophils. Now, in both trials, when we look at the eosinophilic populations only, mepolizumab 100 milligrams appeared to reduce the rate of exacerbations by around 20%. And here we can see the secondary outcomes. Metrics showed an improved time to first moderate or severe exacerbation with no significant impact on pulmonary function or health status, and importantly, no safety signals. In this interesting post hoc analysis, we can see that the higher the eosinophil level, the better response to mepolizumab, as we would expect. And coming to the more recent trials from the Matini trial, this was again a phase 3 trial that recruited 800 patients to mepolizumab every 4 weeks versus placebo, with all the strict eligibility criteria that we would want. So COPD with history of exacerbations, high eosinophils, and patients who were receiving triple therapy at baseline. And we can see that this trial demonstrated again a modest reduction in the annualized rate of moderate or severe exacerbations, approximately 20% reduction, a significant reduction in the rate of severe exacerbations, 35% reduction, and no significant impact on the health-related quality of life or pulmonary function or safety signals. And the last mepolizumab trial that I would like to discuss was the COPD HELP trial, which recruited patients presenting hospitalized with an acute exacerbation with at least one measurement of high eosinophils, at least 300 at any time in the preceding year. with 238 patients that did not show any significant difference in the median time to hospitalization or death, to the next episode of hospitalization or death, but it did show reduced frequency of moderate or severe exacerbations, again 20%, the increase that we have seen in the other mepolizumab trials. I wanted to summarize the results from the anti-alarmin trial as well. So the first two trials, IRIFI 1 and 2, looking at itepekimab and anti-IL-33, were phase 3 trials. We have a press release from the company stating that only one of those managed to meet the primary endpoint, and so the modest reduction in the moderate or severe exacerbations in former smokers, only recruited former smokers for itepekimab, while IRIFI 2 did not meet the primary endpoint. And we have phase 2 trials for tozorakimab, tezepelumab, and tezepelumab that all were negative for their primary outcome, but they all showed promising result in specific subpopulations. And I think the results from all this trial, if we try to interpret them all together, would be that biologics have a role in COPD, but we haven't found the exact population where we need to target them. Here is what I believe may be the reason. So when we look at eosinophils, there are two subtypes of eosinophils: the resident eosinophils that have more homeostatic roles, and the inflammatory eosinophils that are those that cause the inflammatory response in airway diseases. Now, in asthma, most of the eosinophils are inflammatory. So when we use when we measure blood eosinophils in asthma, we mainly refer to inflammatory eosinophils. And higher eosinophils are better associated with treatment response to biologics and, of course, to inhaled steroids as well. In COPD, the story is a bit different. Patients have more resident eosinophils, and perhaps the bulk number of eosinophils that we are getting does not 100% reflect inflammatory eosinophils. And if we were able to have more targeted biomarkers, we might be able to focus our interleukin 5 or 413 to the right pathways. When it comes to the anti-alarmins, the objective of trials have been, both in asthma and COPD, to capture both the T2 and non-T2 inflammation. Now, in COPD, people who have low T2 inflammation also have significant microbial dysbiosis, and they are predisposed to infections. So the question that we need to answer is to what extent anti-alarmins help with inflammation, or in which patients help with inflammation, thus improving outcomes even in people with T2 low inflammation, and to what extent the predisposition to infections may be worse in patients with, in some patients with T2 low inflammation. Another important consideration for me is the time point where we start, where we identify patients. Perhaps most of us, if we had the thought about who are the patients we want to help most, we would go for patients with a lot of exacerbations, who are already suffering a lot from their COPD. Perhaps this is not the right patient group, because the inflammatory signals are a bit more damp in these people at the later stages of disease, first, and second, because the damage that they have in their lungs and also their debilitation mean that they won't gain as much benefit from biologics as we would want. An important thing that I would like to highlight is that none of the trials of the trials that I presented showed significant safety signals. However, they all excluded patients with significant comorbidities. When we use biologics in our real life, we will have more comorbid patients. So we still need safety data in this population, and the real life studies that will start appear in the next months or years will help will inform our management. Now, what we can do in reality, first of all, I have to say that in UK we don't have access to biologics for COPD yet, so I don't have personal clinical experience. I believe that we have to translate our multidisciplinary team approach that we use in asthma to COPD, meaning that before considering biologics, we need to be to optimize treatment adherence, inhaler technique. We have to address comorbidities. We have to explore other treatable traits, including deconditioning, sarcopenia, perhaps breathing pattern disorder. And when we start biologics, we need to be vigilant, especially in comorbid people, for side effects, especially the first period. And we need to be critical around the response, which is very complicated, first of all, because we don't have the responses that we would expect in asthma, but also because COPD is a progressive disease. So again, perhaps not an increase in the rate of exacerbation may already be a win. So as a take-home messages, biologic treatments appear to be safe and effective, but for carefully selected patients with COPD, and I believe we still need better biomarkers and precision medicine approaches to help us maximize the value for money and the safety benefits balance for these medications. Thank you.